So once the B cells and T cells have been activated, they are going to have the challenge of the B and T lymphocytes, which is to proliferate and differentiate. So proliferate means to make many copies, and differentiate means to make different kinds of cells. So subsets of cells are activated, genes are switched so that they are going to turn on certain genes that will cause the cell to become a different subset. So with the B cells, you have three major types: the memory, the plasma cells, and the regulatory cells. And the plasma cells actually make antibodies, which is the response of the B cells, is the humoral immunity. It's the antibodies in the blood. B regulatory actually will regulate what's going on with the T cells, surprisingly. And the T cells have memory cells that are CD4 and then memory cells that are CD8 that are considered part of this challenge of the B and T cells. And then they go on into the response. And the response is different types of T cells. So in this response of the B and T lymphocytes, and this is a lot, so I gave it its own slide, but the plasma cells are going to make antibodies, and you need to be able to list at least 4 of these functions. So agglutination, they clump together; neutralization, they neutralize their target; complement activation, they activate the complement that would usually result in the MAC attack and the lysis of the cells; destruction, target for destruction, they're going to tag cells, and that increases phagocytosis, and that's sort of related to opsonization, and that's related to opsonization, and it's going to be sort of the same thing. You're coating and preparing the microbe or the antigen to be eaten by phagocytes. So it's prepare food to be eaten. That is what opsonization is. That's the Latin word that it comes from, the root. And if you prepare food to be eaten, well, in cells, what is cell eating? That is phagocytosis. And so that is the antibody functions, and we'll talk about those in a little bit more detail later. Again, this is just kind of an overview to show you how things work, and also kind of an assessment of what you know about and if you can explain how these things are related. So this is going to be a test question at the on the unit exam. So CD4 cells are going to be T helper 1, T helper 2, T helper 17. So the T helpers, the T regulatory cells, and the cytotoxic T cells. So those are the 3 main kinds. T helper 1 increases the immune response and activates macrophages. I kind of ran out of room, so ACT is just activate. So, and the upward arrow means increase. So it increases the response, activates macrophages, and then your T helper 2 is going to increase eosinophils for eukaryotic parasites. And T helper 17 is going to increase inflammation. So most of these, like macrophages, are phagocytosis. Inflammation, that's a second line of defense. Eosinophils, second line of defense. And then the activating the B cells, that's the T follicular helper cell. And T follicular helper cell, this is kind of a newer cell type that's in these things called germinal centers, where in the follicles they are going to be closely associated with and will activate B cells and cause them to, with different cytokines, to produce different kinds of antibodies and to produce different types of T cells, so T cell differentiation and growth, and then also different kinds of antibodies are going to be made because of the signals from the T follicular helper cell in the germinal centers of the lymph node or spleen. So that's kind of a newer one. We used to think that the T helper 2 did that, but now we're finding that it's T helper 2 is more associated with these eukaryotic infections, these eukaryotic parasites. So that is now the T follicular helper is the job of the to activate B cells. And the T regulatory is going to dampen the response. So if you have something that increases the response, you have to have something that decreases the response. And then the cytotoxic T cell, which is the only CD8 cell, is going to directly kill. So that is, those 4 slides are going to be what you need to know for a big essay question diagram that you're going to draw on the test. And you'll have basically a page in the front and a page in the back, and you may do this landscape. I think that's probably more appropriate, but every time I do this, I do it slightly differently to try and fit things in there and to be a little neater and more organized on it. But, you know, however you want to arrange things, just make sure that you understand like which parts are the first stage, second stage, third stage, fourth stage. So the lymphocyte development, the presentation of the antigen, the challenge of the B and T cells, and the response of the B and T cells. So that is all for that's overview. You can see this is not something I just made up. This is basically from the book and how they describe the 4 stages: the lymphocyte development, presentation of the antigen, challenge of the B and challenge of the T. Challenge and the B cells make antibodies, and that's their antibody-mediated or humoral immunity, and the cell-mediated is the T cell-mediated immunity. So here again you can see the lymphocytes are developing from the stem cell. They go to the bone marrow to get differentiated. Here the thymus is where the T cells get differentiated, and then they end up in the lymph nodes at various locations, and this is their picture of the presentation of the antigens, and then the challenge of the B and the T lymphocyte, and then the response of the B and T lymphocytes. So the markers on cell surfaces are involved in recognition of self from non-self. And the major functions are attachment to non-self or foreign antigens, and binding of the surface receptors that indicate self, and that's the MHC molecules, receiving and transmitting chemical messages to coordinate the response, and aiding in cellular development. So you have all these receptors on the cell that are going to receive these chemical signals. That's kind of the long and the short of it. So there are 3 classes of MHC genes, which are major histocompatibility markers. So major histocompatibility marker class 1, the genes for class 1 are what are going to be found on all, they're going to produce the markers that appear on all nucleated cells. So this is kind of a key thing here, that it's on all nucleated cells. And so if a cell is nucleated, it has an MHC1 on it. And what this is doing is it's displaying unique characteristics of self. So this is basically your self marker. It allows for recognition of self from the regulation of immune reactions. So it basically is involved in tolerating self-antigens and preventing us from reacting to self-antigens. So each human inherits a particular combination of these MHC1. The only people that would share this is if you have identical twins or identical triplets. And so it's very rare that they're going to have this exact same combination of MHC1 genes. And this is important because, you know, you may be very similar but may not be an exact match. And when they do transplants, they want to find as close of a match as possible. So the only people that can do a transplant without really being concerned of a tissue or transplant rejection would be people who are identical twins. So class 2 MHC genes are found on the antigen presenting cells. So these are on only the antigen presenting cells, which are the macrophages, the dendritic cells, and the B cells. So B cells, and all of these cells, they are nucleated. So they also have MHC1. So the only group of cells that have both MHC1 and 2 would be the antigen presenting cells. So they have to be, you know, marked as self, but they also have this unique job of presenting antigen, and they present the antigen to T helper cells in the MHC class 2. So it's involved in presenting antigens to the T cells, and specifically to the CD4 cells. And so the MHC1 is going to present antigen to CD8 cells. And so I have my little cheer, 2, 4, 1, 8. Who do we appreciate? T cells, T cells. Yay, T cells, right? So 2, 4, 6, 8 is how the cheer typically goes, but I changed that from 2, 4, well, 2, 4 is still the same, but 1, 8 is changed from 6, 8. So 2, 4, 1, 8. So the T helper or CD4 cell is presented antigen by the MHC class 2, and MHC class 1 presents the antigen to CD8s. Now, there is a third class of MHC genes. These are all located on chromosome 6 in humans, and they encode 4 proteins that are involved with the complement. So when we make complement proteins in the liver, and they're secreted and they go into the bloodstream, that is the MHC class 3 genes are found, are what do the code for complement. So MHC1, you can see, are found on all cells, and they have just one little transmembrane. So MHC1 are found on all nucleated cells, all human nucleated cells. And that's what tells you this is Susan's cells, or this is George's cells, or this is Harry's cells, you know, whoever they belong to. And that allows your immune system to tolerate self-antigens. Then this is the antigen-presenting cells will have MHC2 and MHC1 because they are nucleated cell and they are an antigen-presenting cell. So they have this unique MHC class 2 molecule, which has a place for the antigen. There's actually an antigen binding site over here as well for the MHC1. So antigen presenting cells can present antigen on an MHC1 or on an MHC2. So they can actually activate cytotoxic T cells or CD8 cells. They can also activate the T helper cells. So CD means cluster of differentiation. There's like 400 different CD markers that have been found that are involved in immunity. And there are various different functions, but we're just going to learn those two, CD4 and CD8. So you can see there are some others. There's like CD3 here that is on the T cell surface, but T cells have the B cell receptors that have this Y shape. That's basically an antibody. They have an MHC2 and they have an MHC1. So this is not showing the MHC1s, but they have an MHC2 and an MHC1 because they're an antigen presenting cell and a B cell. T cells have the T cell receptor, and then they have these either CD4 or CD8 markers that are in between these little molecules called CD3 markers. And that is what the surfaces of the B and the T cell look like. So the entrance and presentation of the antigen, they ingest pathogens, so the tissue macrophages are going to eat them up, chew them up, spit them out. Go team go, right? So they're going to ingest the pathogens and induce inflammatory response in the tissue if appropriate. The tissue dendritic cells will ingest antigen, migrate to the lymph nodes, and that's where they present the antigen. The antigen presenting cells are dendritic cells, macrophages, and B cells. So the antigen challenge in clonal selection, the B cells and T cells proliferate and differentiate, so they make clones. Clones are exact identical copies, but some of them will express differently. They'll be differentiated differently, but they have the same antigen that they're going to be made for. Their T and B cell markers are the same. They just may be differentiated into different kinds of cells. So that's the B and the T cells. They proliferate and differentiate. They make many copies and then they differentiate into different kinds. So the T cells are involved in the T cell response, and so they have 3 main functional types of cells. They have helper cells that activate macrophages, and assist B cell processes, and they help activate T cells that are cytotoxic T cells, but they basically activate. Regulatory suppress. So they used to be called suppressor cells, but they control the T cell response. And so if you have something that activates, you have to have something that deactivates or suppresses. The cytotoxic T cell then leads to the destruction of the infected host cell or other foreign cells, and basically they blow them up. So they have perforins and granzymes, and they basically lyse the cells. And so those are our three types, and that's the cell-mediated immunity. Well, the B cells make plasma cells, and the plasma cells release antibodies, and so this is the humoral immunity. And so they have different jobs. They have marking for destruction, neutralization, agglutination, all the things that we talked about. So that's a summary of what was on the chart earlier. So concept check for section 15.1. The antigens are usually blank or blank molecules. See if you can answer that question. Number 2, maturation of B cells occurs in the blank, and maturation of the T cells occurs in the blank. Question 3, MHC markers are on all cell types except what? And 4, dendritic macrophages and B cells all serve as blank, blank, blank. And question 5, true or false, T cells produce antibodies. And question 6, true or false, when activated, both B and T cells produce memory cells. And we'll start with the answers in the next section, the next video.